audible or not? Yes, please respond.
Audible, sir.
Yes, very good afternoon. Is it audible or not?
Yes, sir. Audible, sir.
Okay.
Okay, no, I'm not talking.
Okay. Today we will discuss about structure and function of insect integument. Structure and function of insect integument. So it will come under insect morphology course. So that's why I want to learn about the definition of insect morphology. So insect morphology means it is the study and description of the physical form of an insect. Study and description of the physical form of an insect. So physical means external characters of an insect. So it includes antenna. You can see here antenna, mouth parts, eyes. So different types of legs is there, and wings is there. So it will all come under external characters of an insect. It will come under insect morphology. So next, the famous scientist, so the father of insect morphology. Father of insect morphology. So R.E. Snodgrass. R.E. Snodgrass. So means Robert Evans Snodgrass. Robert Evans Snodgrass. So he wrote famous book that is the Principles of Insect Morphology. Principles of Insect Morphology was written by R.E. Snodgrass. So next one, the famous book, The Insect Structure and Function of an Insect. Structure and Function. The Insect Structure and Function. The book was written by R.F. Chapman. So that's all about introduction of insect morphology. It includes the different types of external characters of an insect. So let's move on today's topic, structure and function of insect integument. Structure and function of insect integument. So integument we can also call as body wall. Integument or body wall or exoskeleton. So whatever it may be, same name. So in case of ICAR examination, so sometimes they will mention body wall, sometimes they will mention exoskeleton, sometimes they will mention integument. So don't confuse with those names. So both are same only, integument or body wall or exoskeleton. So integument of an insect, which can form an exoskeletal covering of the insect body. It means externally it can cover the insect body. So that's why we want to call as exoskeleton. So if it is inside, we want to call as endoskeleton. So endoskeleton, that is different. So in case of tentorium, insect head, endoskeleton is there. But in case of externally, exoskeleton is there. So that's why I want to call as insect integument or body wall or exoskeleton. So these integuments, so it can form a skeleton of the insect body. So it can act as a skeleton of the body. So these insect integuments, so it can also provide some area for muscle attachment. So it can also give some area for muscle attachment to these body walls. So it can also give protection from desiccation. Protection from desiccation. So in case of dry conditions, evaporation it will take place. So at that time, insect it want to conserve some moisture. So at that time, these body wall or integument, it can give protection against protection from desiccation. So in case of physical or mechanical injuries, and it will also give some shape to the insect body. So even you can imagine our body. So because of these body wall, so it can give some shape to human beings. So like that only insect, so it can provide some shape, strength. Okay. So these integument or exoskeleton or body wall, so it's divided into three types, or it consists of three parts. Those are inner basement membrane, middle hypodermis, inner basement membrane, middle hypodermis, and outer layer cuticle. outer layer cuticle. So these exoskeleton or body wall or integument, whatever it may be, it consists of three parts. Those are inner basement membrane, middle hypodermis, and last one, outer covering, cuticle. The inner basement membrane, it is the starting layer, basal part of the body wall. So hypodermis means unicellular is there. So remaining two are okay, very good afternoon. So today we'll discuss about structure and function of insect integument. Structure and function of insect integument. So it will come under insect morphology. So that's why we want to learn about the definition of insect morphology. So insect morphology means the study and description of the physical form of an insect. Study and distribution of a description of the physical form of an insect. So physical form, it means external character of an insect. External characters, it includes antenna, eyes, mouth parts, and wings, different types of legs. So it will all comes under different types of. So it will all comes under external characters of an insect. So that is the definition of insect morphology. So next, the famous scientist, the father of insect morphology. The father of insect morphology. So R.E. Snodgrass. So R.E. Snodgrass. So R.E. means Robert Evans Snodgrass. So he wrote the famous book that is the Principles of Insect Morphology. Principles of Insect Morphology. So another the famous book that is the Insect, the Structure and Function. This book was written by R.F. Schaffner. So that's all about introduction of insect morphology. Let's move on to today's topic, structure and function of insect integument. Structure and function of insect integument. So integument we can also call as body wall or exoskeleton. Insect integument we can also call as body wall or exoskeleton because it can cover externally. So exo means outside. So it can cover outside of the body. So that's why we want to call as exoskeleton of the body. So endoskeleton, so that is different. So that is called a centurium. So endoskeleton is different, exoskeleton is different. So exoskeleton means external covering of the insect body. So that's why I want to call as exoskeleton of the body. So these integument of an insect, so it can form the skeleton of the insect body. So it will also provide some area for muscle attachment. So it can provide some area for muscle attachment. So it can also give protection from desiccation. So in case of dry conditions, so at that time evaporation is there. So for control of those evaporation, these insect cuticle or integument, so it play a major role. So it protection from the desiccation. So in case of physical and mechanical injuries, it will also give some shape to the insect. So in case of human body, so this body wall, it will also provide some shape to human beings. So like that only insect, so it can give some shape and strength to the insect. So these exoskeleton or integument or body wall consists of three layers. So those are inner basement membrane and middle hypodermis, and third one outermost cuticle layer. So inner basement membrane, middle hypodermis, and outermost cuticle. So inner basement membrane, so basal part of the body wall. Hypodermis we can also call as epidermis. Hypodermis we can also call as epidermis. So cuticle, there are two types of different cuticle is differentiated into two types, epicuticle and procuticle is there. So anyway, we will discuss later. So first we will discuss about inner basement membrane. Inner basement membrane. So you can see this is the diagrammatic representation of the insect body wall. So you can see here. So this is the innermost layer, basement membrane layer. So middle epidermis or hypodermis layer. This is the middle layer. Epidermis or hypodermis. And remaining portion, it is the outermost layer we want to call as cuticle. So cuticle is differentiated into two types, epicuticle and procuticle. Epicuticle and procuticle. So you can see here, so these are the unicellular, means epidermis or hypodermis are unicellular in layer. But in case of basement membrane and cuticle are non-cellular, means there is no involvement of cells. So cells are absent. So you can see here, basement membrane, it means basal part of the body wall. Basal part of the body wall. So which are formed from the degenerated epidermis cells. So which are formed from degenerated epidermis cells. So at the time of molting, some epidermis cells, it will be degenerate. So from those degenerated epidermis cells, so these basement membrane will form. So it is appear as a shapeless, means non-living. So there is no involvement of living cells. So it's a non-living layer. So that's why there is no shape. So the thickness of basement membrane is 0.5 microns in size. Thickness of basement membrane is 0.5 microns. So it is also consists of fibrous proteins materials and glycosaminoglycans, which are polymers of disaccharides. So different types of saccharides is there, monosaccharides, disaccharides, polysaccharides. But these glycosaminoglycans are disaccharides. So it is also non-cellular membrane. So non-cellular membrane, basement membrane and cuticular membrane are non-cellular membrane. So it can underlying the epidermal cells of the integument. So next, second layer, the epidermis. The second layer, epidermis. So I already discussed about it is the unicellular layer. So I already told that point that is the unicellular, because uni means only one cell is there. So unicellular layer and formed from the polygonal cells. So which are formed from polygonal cells. So you can see this picture. So unicellular. Cytoplasmic epidermis or epidermis layer. So this is another cell. So these two cells are held together by the desmosome. So another cytoplasm that is called as desmosome. So adjacent epidermal cells, which are held together by means of certain cytoplasmic processes, we want to call as a desmosome. So in case of these epidermal cells, so which can secrete, it can act as glandular and it can also secrete the cuticles. So it can secrete the cuticles. So it can also secrete the cuticles. So at the type of molting process, so these polygonal cells, which can modified into cuboidal or columnar cells. So these epidermal cells get differentiated into 5 types. Those are dermal gland, trichogen cell, molting gland, peristigmatic gland, and oocytes. Dermal glands, trichogen cell, molting glands, peristigmatic glands, oocytes. So derma means skin. So it can produce in the cement layer. It can secrete the cement layer. So trichogen cell. So there are 2 types of cells, is the trichogen and tarmogen. So we'll discuss later about these type of cells. So trichogen cell means hair-like setae. So it can hair-like setae, trichome, nothing but hairs. So in case of our body, different types of hairs is there. So in case of head region, so one type of hairs is there. So like that only insect body covered with hairs. But in case of entomology point of view, we want to call as a trichomes, trichogen cells. So next, molting glands, it can secrete the molting fluids. So molting glands, they can secrete the molting fluids at the time of molting process. So they can digest the old cuticles and they can produce the new cuticles. And peristigmatic glands, you can remember this point, peristigmatic glands. So it can present around the spiracles of dipterans larva. So it can produce around the spiracles. So in case of respiratory systems, so different types of spiracles is 10 pairs of spiracles is there. So at that time, respiration. So in case of dipterans larva, so these spiracles contain peristigmatic glands. So these peristigmatic glands are present in spiracles of dipterans larva. So another one, fifth one, enocytes. So enocytes. So it can produce the lipoproteins. So lipoproteins is there, so which are deposited by epidermal cells. So lipoproteins, enocytes, lipocytes, which are deposited by the epidermal cells to form a protein epicuticle. So protein epicuticle means cuticle layer. So in case of cuticle, different types of layers is there, epicuticle, procuticle. So there it will come under protein epicuticle. So cuticle layer we will also call as protein epicuticle. You can see here it is a diagrammatic representation of insect integument. So innermost basement membrane is the epidermis or hypodermis unicellular, and remaining are cuticle only. So upper epicuticle and procuticle is there. So let's move on. Third one, cuticle. So cuticle, it was divided into two types. Those are upper epicuticle and inner procuticle. So cuticle, it is the outermost thick layer, outermost layer of the integument, outermost, innermost basement membrane, outermost cuticle only. So it is secreted by the epidermis only. So at the time of molting process, so it can secrete the cuticle. So both basement membrane and cuticle is secreted by the epidermal cells only. But the epidermal cells, they can secrete from the polygonal cells. Polygonal cells, they can modify into cuboidal or columnar cells at the time of molding process. So first we will discuss about epicuticle. So this epicuticle is so thin outermost layer. Outermost layer, it is a thin, it is a varying thickness varying from 1 to 4 microns in size. The thickness of epicuticle is 1 to 4 microns. The most important point is chitin is absent. Chitin is absent in epicuticle. So in case of ICR examinations, I think in 2018 they asked this item. So chitin is absent in which cuticle, epicuticle or procuticle? So epicuticle, chitin is absent. So it's a very, very important point. So very, very important bit also. So chitin is absent in epicuticle. So it consists of 4 layers. So those are tectocuticles. Tectocuticle we will also call a cement layer. So sometimes they will ask a tectocuticle, sometimes they will ask a cement layer. So cement layer is nothing but tectocuticle. So like that only wax layer. So like that only wax layer. So wax we will also call as lipid epicuticle. Wax layer we will also call as lipid epicuticle. So protein epicuticle is cuticle layer. Protein epicuticle is cuticle layer. Lipid epicuticle is wax layer. You can remember those terminology. So third one, polyphenol layers. So polyphenol layers. So tectocuticle means cement layers. So dermal glands, they can secrete the cement layers. Dermal glands, they can secrete the cement layers. So cement means outermost layer. So it can give protection against the damages, external damages. If it is any external damages, then cement layer, it can give protection to those insect bodies. So next one, wax layer. The wax layer we will also call as lipid epicuticle. Lipid epicuticle. Wax layer we will also call as lipid epicuticle. So these wax layers, so it consists of esters of fatty acids, is there alcohols. The thickness of a wax layer is 0.25 microns in size. So it can act as a waterproof. So wax means it can act as a waterproof. So polyphenol layers. Polyphenol means it is a non-static layer. So it's resistant to acids, organic solvents. So if you apply any chemical, so then it can insect will resistant to these polyphenol layers only. So next one, cuticle layer. Cuticle layer means it serves as the purpose of permeability. So insect stretchability, it will depend on this cuticle layer. So we will also call as protein epicuticle. Protein epicuticle, we will also call as cuticle layer. So first we will discuss about epicuticle. You can see these methods. We already discussed about this method. So it is the outermost layer. Thickness varies from 1 to 4 microns in size. Thickness, and chitin is absent. It's very, very important. So chitin is absent. So next, pore canals. So pore canals, it is the size of pore canal. Pore canal is less than 1 micrometer. The size of pore canal is less than 1 micrometer. Less than 1 micron. It is present in the exocuticle, which helps in the deposition of epicuticle only. So pore canals which are present in the exocuticle. So epicuticle, procuticle. Procuticle again divided into two types, exocuticle and endocuticle. So in case of those exocuticle, the pore canals are present. These pore canals, it can helps in the deposition of epicuticle. So it consists of 4 layers. So those are cement layer, wax layer, polyphenol layer, and cuticle layer. So cement layers we already discussed about these points. It is secreted by the dermal glands. So dermal glands is secreted by dermal glands and is composed of helicoprotein is also. The function of cement layer is it can give protection to the body from external damage. So next, wax layer. The thickness of a wax layer is 0.25 microns. 0.24 microns. It also consists of a long chain hydrocarbons, esters of fatty acids and alcohols. So the function of wax layer is the waterproof layer. So waterproof layer, so preventing water loss from the body, desiccation. So next, polyphenol layer. Polyphenol means non-static layer. So it's containing of various types of phenols. So it is the resistance to acids and organic solvents. The function of polyphenol layer is resistance to acids and organic solvents. Next, cuticle layer. Cuticle layer. So it's an amber color. So thin layer over the surface of the epidermis. So which is strengthened by outer polyphenol layers. So the main function of cuticle is the purpose of permeability. So it can also act as a growth barrier. It can also act as growth barrier. So these are some points which are related to epicuticle. Epicuticle. So next one, procuticle. Upper epicuticle over. Next, inner procuticle. So it was divided into two types. Those are exocuticle and endocuticle. So exocuticle, endocuticle. So exocuticle, it is darkly pigmented. The exocuticle means darkly pigmented, darker in color because melanin contains more. So in case of darkly pigmented means melanin contains more. So it is also hard and sclerotized. So exocuticle means darkly pigmented, but in case of endocuticle, soft in color, softly pigmented. So soft in color, non-sclerotized. So it can give the rigidity to the cuticle. So in case of these exocuticles, it can give rigidity to the cuticle, and it also consists of mainly chitin. So mainly chitin, and one protein is there, that is the sclerotin. Sclerotin, it is present in exocuticle. Arthropodin, it is present in endocuticle. So you can see here exocuticle. So it is a highly darkly pigmented color and hard and sclerotized pasta. So it gives rigidity to the cuticle, rigidity, and it also consists of mainly chitin. So mainly chitin, it is present in exocuticle, and the hard protein, that is the sclerotin. Sclerotin, it is present in exocuticle. Arthropodin, it is present in endocuticle. The outer layer, it is the outer layer, much thicker with composition of chitin and sclerotin. So this layer is dark in color and rigid is also there. And this sclerotin, it is the protein. Sclerotin, it is the protein, is the water insoluble because it's a tanned protein. Because it is a tanned protein, so that's why it is water insoluble. But arthropodin is water soluble but untanned protein. So arthropodin is different, sclerotin is different. Don't confuse with this terminology. Sclerotin means water insoluble, so but tanned protein. But in case of arthropodin, it is a water soluble but untanned protein. So you can see here, so darkly pigmented. So this is the exoskeleton, exocuticle. So it is the exocuticle. So darkly pigmented, but in case of endocuticle, lightly pigmented, soft in color. So you can see here endocuticle. So it is soft and light colored, but in case of exocuticle, dark and hard and sclerotized. So it is unsclerotized, quite opposite to exocuticle. So exocuticle, it is hard, darkly pigmented and sclerotized, but endocuticle is quite different. So it is also consists of more chitin. So in case of ICR examination, so they will ask this type of bit. So more chitin is present in which cuticle? Endocuticle. So abundant quantity of chitin is there. So but it's lack a hard protein, that is sclerotin. So that's why it is a soft and light colored. If sclerotin is present, then this cuticle will become hard and dark colored. So it's compared to other, it is the inner and thickest layer is also. So it is made up of chitin and arthropodin. But in case of exocuticle, chitin and sclerotin. But in case of endocuticle, chitin and arthropodin. So these arthropodin is the water soluble, but it is an untanned protein. So untanned, so that's why it is soluble in a water. So these arthropodin, it is also constitute 25 to 37% of the cuticle. So if it is cuticle 100%, if you consider as 100%, then these arthropodin, it constitutes 25 to 37% of the cuticle. Okay, so tanning, it is a process where arthropodin, it is converted into the sclerotin. So tanning, it is done process. So arthropodin means in case of endocuticle, arthropodin is present, which later it will converted into the sclerotin. So in case of at the time of molting, first they can secrete soft color, soft in color. So later it will turn into the darker. So that's why arthropodin is converted into sclerotin. That process we want to call it the tanning. So tanning is mostly bring by the protein, so that is the quinone protein. So quinone protein, it can bring the tanning. So it's derived from the tyrosine, that is the amino acid. One protein is there, so that is the quinine protein. So like that only amino acid, tyrosine. So one hormone is involved in this tanning process, that is the Busk-Con, not Busk-Cor, Busk-Con hormone, that is Busk-Con hormone. So with the help of these hormones, tanning process will take place. And protein is also helpful in this process, that is the quinone protein, and tyrosine, it is the amino acid. Okay. So next, pore canals. On pore canals is also there. The size of the pore canal is less than 1 micron. So you can remember those ones. So these pore canal, it is present in exocuticle only. So that is present in exocuticle, but it can secrete the cuticle layer or epicuticular part. Okay. So next, characters of the cuticle. So it consists of two major characters. So those are chitin and protein. So cuticle, it consists of two major characters. So those are chitin and proteins. So chitin, first we will discuss about chitin. So it's a nitrogenous polysaccharide. But in case of a basement membrane, so glycosaminoglycans, it is a disaccharide. But it's a nitrogenous polysaccharide. Chitin is a polysaccharide. But in case of basement membrane, there disaccharides is there. So it is the nitrogenous polysaccharide. The chemical formula of chitin is C8H13O6N. So I think in last year, in 2019, in case of ICR examinations in IRA entrance, so they asked this question. So what is the chemical formula of chitin? C8H13O6N. So that is the chemical formula of chitin. You can remember this one. So that chemical formula, this chitin was named by Oder. So in 1835, so the chitin was named by Oder. It is the high molecular weight. It consists of high molecular weight polymer of N-acetyl glucosamine residues. So these are joined by beta glycoside linkages. So it is also very, very important bit. So I think in 2017 they asked this bit. So N-acetyl glucosamine residues. So chitin, it belongs to chitin character. It consists of high molecular weight character. High molecular weight polymer of N-acetyl glucosamine residues. So it is a nitrogenous polysaccharide made up of chains of N-acetyl glucosamine and glycosamine units. So in case of N-acetyl glucosamine, 6 is there, but like that only glucosamines 1 ratio is there. So these are linked by beta glycosidic linkages. So beta 1,4 beta glycosidic linkages. It is insoluble in water. So it is insoluble in water, alcohols or organic solvents, dilute acids or concentrated alkanes, but soluble only concentrated minerals are sodium hypochlorite. So chitin, which is soluble in sodium hypochlorite. The specific gravity of chitin is 1.4 specific gravity of chitin is 1.4. Like that only chitin stretchability. So that is 13 mm. Chitin stretchability is 13 mm. So different types of chitins is there: alpha chitin, beta chitin, gamma chitin. So among those chitins, alpha chitin is most stable component which is present in all the portions. Alpha chitin. So in case of ICR examinations, they will ask this type of beta. So which type of chitin? Alpha, beta, gamma. So only alpha chitin, which is a stable component present in all the portions. Okay, you can see this is the structural formula of chitin. Structural formula of chitin. Next one, proteins. Second character, protein. So in case of proteins, there are 3 types of proteins is there. Those are arthropodin, sclerotin, and resilin. Arthropodin, sclerotin, and resilin. So arthropodin, it is present in endocuticle. Sclerotin, it is present in exocuticle. So resilin, it is rubber-like elastic protein. So rubber-like elastic protein. So which is a it is present in okay, sorry for the inconvenience. Okay, sclerotin. So with the help of Buscon hormone, so this tanning process will take place. So that's why we'll also call as tanned protein, which is amber in color and present only in the exocuticle, which is only present in the exocuticle. So resilin means next one, third protein, resilin. So it is a rubber-like elastic protein, so which is present in ligaments, so wing ligaments, or leg joints, or clipolabral joints. So in case of head regions, or different types of sutures and sclera existed. So at that time, so these resilin it is present. So next one, so outgrowth are internal imaginations of the body. It's a very, very important topic. So cuticle or integument or body wall or exoskeleton. So imaginations are external outgrowth of the body wall. So you can see it is the integument or body wall or exoskeleton. So it consists of 2 types. So those are outgrowth, cuticular outgrowth and cuticular invaginations. So cuticular outgrowth and cuticular invaginations. So these cuticular outgrowths again divided into 2 types. Those are cuticular appendages and cuticular process. So cuticular appendages and cuticular process. So like that only cuticular invaginations, it will consist of 2 types. Those are apodeme and apophysis. Apodeme and apophysis. So again, these cuticular appendages consist of 2 types. Those are setae or macrotrachea and spur. Setae or macrotrachea or spur. So cuticular process, like that only, it consists of 2 types. Those are microtrachea and spines. Microtrachea and spines. So these body wall are first we will discuss about these cuticular outgrowths. So it was divided into 2 types. So those are cuticular appendages and cuticular process based on the presence of membranous articulations. So presence of membranous articulations. So these cuticular outgrowth was divided into 2 types. Those are cuticular appendages and cuticular process. So you can see first we will discuss about these cuticular appendages. Cuticular appendages. So it consists of 2 types. Those are macrotrachea. We can also call as setae. So like that only another one, spur. So cuticular appendages. So these are the outgrowth of the cuticulum. So our outgrowth of the cuticle or integument. So it is connected with it by means of membranous joints only. So these arise from the modified epidermal cells. So these are produced from the modified epidermal cells. So cuticular appendages means externally. So it is visible to our eyes. It is visible to our eyes only. So if you observe any insect with the help of our eyes, so that is the cuticular appendages, means external covering, outgrowth of the insect, cuticle, outgrowth. So like trichomes, hairs. So this consists of setae and macrotrachea or sphagnum. So first we will discuss about these setae or macrotrachea. So these setae are macrotrachea. So it consists of 4 types of plants is there. So 4 kinds of setae is there. Those are clothing heads, scales, glandular setae, sensory setae. Setae are macrotrachea. Again consists of following types. Those are clothing heads, scales, glandular setae, sensory setae. So in these setae, so trichogen is there, Tarmogen is there. Trichogen is there, Tarmogen is there. And the study of arrangement of these setae we want to call as ketotaxy. It's a very, very important bit. Study of arrangement of these setae means different types of setae is there. So clothing heads, it is also one type of setae. Scales, it is also one type of setae. Glandular setae, sensory setae. So study of these type of setae we want to call as ketotaxy. We want to call as ketotaxy. And trichogen cell is also there. Trichogen cell. So single epidermal cells. So when larger of single epidermal cells, that is called as trichogen cell. And side by side second hypodermal cells we want to call as Tarmogen cell. So you can see here the cuticular outgrowth. We already discussed these points, which was divided into 2 types. Those are cuticular appendages and cuticular process. Based on the presence or absence of membranous articulations, so these cuticular outgrowth was divided into 2 types. Those are cuticular appendages and cuticular process. So first we will discuss about cuticular process. So it is the outgrowth of the cuticle. Means externally it is visible to our eyes. So it's arises from the modified epidermal cells. So it is classified into 2 types. Those are setae or macrotrachea or sphagnum. Second one, sphagnum. So we will commonly call as hairs only. So which is arises from the cup-like alveolar surface. We want to call as trichogen. So you can see here. So the setae are hollow structures. So means empty within those trachea is empty only. So these setae are hollow structures developed as extension of the exocuticle. So exocuticle is there, exocuticle, endocuticle. So in case of procuticle, exocuticle, endocuticle is there. So at that time, these exocuticle, so it is the extension of exocuticle. Means trichogenil is the extension of exocuticle. And it is also produced by single enlarged hypodermal cells. Hypodermal is nothing but epidermal cells. So that is called as trichogen cell. And articular means side by side. So this articulates the membrane. So that is usually produced by the second hypodermal cell. So that is called as Tarmogen. So in between the exocuticle and endocuticle, one more layer is also there. So that is called as mesocuticulum. So in between the exocuticle and endocuticle, so mesocuticle also there. So mesocuticle. So it is present in between exocuticle and endocuticle. So in case of cuticle, epicuticle, upper epicuticle, inner procuticle is there. So epicuticle it consists of 4 types of cells. So like that only procuticle. It consists of 3 types of cells. Those are exocuticle, mesocuticle, and endocuticle. So, but in case of mesocuticle, so not fully developed darker layer. Not fully developed, not fully darker layer. So darker means melanin content is not there. So like that only chitin. So those chitin constitute nearly 25 to 40% of the dry weight of an insect. So 25 to 40% of dry weight of an insect. So in case of honeybees, cement layer means dermal glands, they can secrete the cement layer. So in case of honeybees, those cement layer is absent. You can see here, study of arrangement of setae we will call as ketotoxy. Study of arrangement of setae we will call as ketotoxy. So in next, different types of setae is there. So those are clothing aids, scales, glandular setae, sensory setae. So scales in the sense, you can see here, scale setae. Scales means butterflies. So clothing heads means honeybees or bees. So in case of those insects, it contains the flopping head. So like that only in case of butterflies, scale insects is there, scale wings. So glandular setae, in case of caterpillars, glandular setae is there. So sensory means it can sense some. So with the help of those setae, it can sense. Insects will sense some. So these are the setae or trichomes, so provided with the sensory receptors, sensory nerves. So next one, cuticular processes. Cuticular appendages or outgrowths over. So cuticular processes. So it consists of two types. Those are microtrachea and spines. So microtrachea and spines. So macrotrachea we can also call as setae. But in case of microtrachea, it will come under cuticular processes. So here membranous articulation is absent. If membranous articulation is present, it will goes to cuticular appendages. If membranous articulation is absent, so it will come under cuticular process. So it consists of two types, microtrachea and spines. So you can see here, so no membranous articulation in case of cuticular processes. So microtrachea we can also call as fixed hairs or equilae. So these are the minute hair-like structures, so which is present on the wings of Mecoptera and certain diptera and so on. So like that only spines. So spines means immobile. So multicellular only, but it's immobile. So multicellular, if it is mobile, then we will call as spur. So multicellular, but if it is immobile, then we want to call as spines. If it is mobile, spur. If it is not mobile or immobile, we will call as spines. Multicellular. So these are multicellular. So outgrowth of the cuticle, which are more or less thorn-like in form, so which are immobile. Spines are immobile but multicellular. So you can see here it is the diagrammatic representation of trachea. Trachea, trichogen, tarmogen, different types of cells. So next, functions of the body wall. So it can form different types of functions. It can perform different types of functions in body insects. Those are it can act as external or more, strengthen the external organs like jaws and ovipositor. So it can also protect the organs against the physical aberration or injurious chemicals or parasites or predators and pathogens. So it can give protection, this body wall, it can give protection. So it can form the body. So internally it can protect the visual organs or vital organs in case of foregut, midgut, hindgut or trachea. So these are the some functions. So it can useful to insects. So it can also prevent water loss from the body. The cuticular sensory organ helps in the sensing the environment. Okay, next one, molting process. Molting process. So molting, it's a very, very important topic, molting. So molting, it means in case of insect cuticle, so it is very hard in structure. So very hard, and it can form the unstretchable exoskeleton. So there is no stretching. So it must be shed from time to time. So in case of molting, at the time of molting, so instars, different types of instars is there. So maximum in case of insects, 5 instars is there. So from one instar to another instar, so molting will take place. So from egg to larva, molting is there. From larva to pupa, molting is there. From pupa to adult, molting is there. So that is called as molting. So from development, at the time of development, they can shed their cuticles at their body wall. So best example, snakes. Snakes are my best examples. So in case of those insect cuticles, so it is very hard and it can form the unstretchable exoskeleton. And it must be shed from time to time. So it permits the insects to increase their size during growth development. So during growth period, so it can increase their size from time to time. So that is called as molting. So that process we want to call as molting process. So periodical process of shedding the old cuticle, which is accompanied by formation of new cuticle. So that is also we want we want to call as molting or egg dices. So periodical process, step by step process, periodical, so one by one step. So shedding the old cuticle and formation of new cuticle we will call as molting. And casting of cuticle we will also call as molting. So you can remember these points also. So molting, there are three definitions is there. Those are insect cuticle which is hard and forms unstretchable exoskeleton, So it must be shed from time to time. So that is also cuticle molting only. So periodical process of shedding the old cuticle, so we can also call as molting. So casting of cuticle, we will also call as molting. So, but stadium, it is different. Stadium means time between two molds. So two intermolds. So from one instar to second instar. So that time period we want to call as stadium. So form of the insect during stadium we will call as instar. So that form insect which will form after molding it can form some shape with the help of these cuticles. So that is called as instar. So don't confuse with this terminology. So stadium is different, instar is different. So instar means form of the insect during stadium. So that is called as instar. And cast of cuticle of an insect we will call as exuvum. So like excreta. So exuvum means cast of cuticles. After molting it can release some cuticles. So that is called as exuva. So this molting process it consists of 3 steps. Those are apolysis, ecdysis, and sclerotization. So the name itself indicating the answer. Apo. So apo means formation. Lysis means dissolution. So the dissolution of old cuticle and formation of new one we will call as apolysis. So apolysis. So one process is there in case of apolysis. So first it can start with the repeated mitotic divisions of the epidermal cells. So in case of body, epidermis is there. In case of body wall, epidermis or hypodermal cells is there. So polygonal cells. So there it can start the mitosis. So with the help of mitotic division of epidermal cells, so the cells increase in number. So the cells increase in number. So in case of mitosis, so 4 cells, 2 cells of 4 cells. But in case of meiosis, 8 cells is there. So with the help of these repeated mitotic division of epidermal cells, so resulting in increase in number and size of the epidermis also. So which become columnar in shape. For polygonal cells, they can modify it into columnar or cuboidal in shape. So because of these increase or because of these changes, the epidermal cells exert tension on cuticular surfaces. So because of these pressures, so side by side, number is increasing. At that time, space is very less space is there. So at that time, because of these epidermal cells, it can exert tension of cuticular surfaces. So as a result, it can get separated from themselves from the cuticle. So from it can separate due to separation of these epidermis from the cuticle, the subcuticular space is created. So automatically it can create some space. So because of those pieces, due to this separation of the epidermis from the cuticle, they cut the subcuticular space is created. The subcuticular space is created. So in their subcuticular space, these epidermal cells they can secrete the molten fluid. They can secrete the molten fluid, so which is a glandular and gelatinous and contains 2 enzymes. So that is the proteinase and chitinase. So you can remember these 2 enzymes. It's a very, very important enzyme. So in case of ICR examinations, so they will ask this type of bit only. So we have proteinase and chitinase. So only these 2 types of enzymes will secrete in the molten fluids. So molten fluids, they can secrete only 2 enzymes. Those are proteinase and chitinase. So with the help of those enzymes, proteinase and chitinase, they can digest the old cuticle. So mid layer, so that is the zone of deposition of new cuticle or procuticle. So zone of deposition of new cuticle or procuticle. So the ordering of deposition of cuticle is first it can form the cuticle layer. So cuticle remains upper epicuticle, inner procuticle, epicuticle and procuticle. So in case of epicuticle, 4 types of layers is there. Those are cement layer, wax layer, polyphenol layer, and cuticle layer. So at the time of molting process, first cuticle layer will form. So first cuticle layer it will form. So first cuticle layer only. Then procuticle. So procuticle contain exocuticle and endocuticle. So procuticle contain exocuticle and endocuticle. Then side by side wax layer, cement layer. So from bottom to top. But first formation layer is the cuticle layer. You can remember this one. First cuticle layer, then followed by exocuticle, endocuticle, then wax layer and cement layer. So this is the order of deposition of cuticle. Order of deposition of cuticle. So next one, ecdysis. So ecdysis means the stage where the insect has both newly formed epicuticle and procuticle. So newly formed epicuticle as well as procuticle and old exo and epicuticle is also there. So that is called as Farett instars. So Farett instars. So that process we want to call as ecdysis. So the stage where the old one and new one. So old one and new one we want to call as Farett instars. So Farett instars. So these ecdysial membranes, so it can start splitting along the line of weaknesses. So due to some muscular activity or some with the pressure of blood, some pressure on the circulatory system. So with the help of circulatory system, so it can create some pressure. It can create some pressure. So due to muscular activity of the inner developing insects, it also because of swallowing of air and water. So resulting in the distension of the gut. So finally, shedding of the old cuticle accompanied by formation of new cuticle we want to call as molting or ecdysis. So the hormones which is responsible for molting is ecdysone. Hormone which is responsible for molting is ecdysone. And cholesterol which can act as a precursor for synthesis of ecdysone. So cholesterol it can act as a precursor for the synthesis of ecdysone. So like that only in case of caterpillars. So Wessner's gland. So Watson's gland. Wesnow gland, it is different. In case of honeybees, it is present. So in case of honeybee pheromones, it can present. Wesnow gland is different. Watson's gland is different. Watson's gland, it is present in the caterpillars. And finally, sclerotization. Finally, last one, sclerotization. So sclerotization means the darking. So in case of the starting stage, so soft color is there, milky substances. So later it will be hard. So that's why we want to call as hard-like structures. We want to call as sclerotization. If it is dark, we want to call as melanization. So the cuticle, it will become so dark. Dark means melanin content. Even some human beings, they will be more darker because of this melanin content only. So cuticle becomes dark, that is called as melanization. If it is hard, we want to call it sclerotization. After shedding of old cuticle, now cuticle which is soft, milky, white colored, so which becomes dark and hard through the process tanning. So Buckskin, with the help of Buckskin hormone, tanning which will take place. So the maximum sclerotization which is found in the mesonotum of adultum. So in case of thorax, prothorax, mesothorax, metathorax is there. So dorsal surface we want to call as notum. Dorsal, tergum, sternum, pleura is there. So like that only notum. So mesonotum means mesothorax. So at that region, so maximum sclerotization is found. So like that only in case of mandibles of larva. So mandibles, so in case of mouth parts, so upper labrum, labium is there, mandibles. So these mandibles also maximum sclerotization is there. So the hormone which is responsible for tanning and sclerotization is Buckskin hormone. So you can see this process. Sequence of steps in hardening of the cuticulum. Sequence of steps. First, tyrosine. So it will form into dopa dihydroxyphenylalanine. So first tyrosine, it will form into dopa dihydroxyphenylalanine. So later it will form into dopamine. So later it will form into dopamine. It is a chemical process. So later it will enter into N-acetyl dopamine. N-acetyl dopamine. So N-acetyl dopamine, it will enter into the N-acetyl dopamine phenone. So N-acetyl dopamine phenone. So finally tanning takes place. So this is a continuous process. So tyrosine, from these tyrosine, it will produce dopa dihydroxyphenylalanine. So it will enter into dopamine. From dopamine, N-acetyl dopamine is produced. So from this one, N-acetyl dopamine phenone is there. So finally tanning takes place with the help of Buckskin. So that's all about today's lecture. So because of some network issue or some issues, today's class or some disturbance is there. Sorry for those inconvenience. So if you have any doubt, you can ask now. Regarding of these. Yes, you can ask.
So can you share this PPT with us?
No. Why you need this PPT?
I was not able to note some of the points, sir.
So I will refer some books, then you can study those books.
Okay, sir.
Sir,
Yes, yes.
can you show the Slide of sclerotization.
Sclerotization.
Yes, sir.
You can see here. So teneral, it is the newly emerged pale soft-bodied individuals. So that's why I want to call as teneral. So in case of sclerotization, first they can secrete milky substances. So that is called as teneral. After completion, it can turn into the melanization or sclerotization. If it is dark, we want to call as melanization. If it is hard, we want to call as sclerotization. So in case of starting, proteinase and chitinase is there. So those enzymes, it can digest the old cuticles. At that time, it can secrete milky substances. So those are white in color. So that's a soft-bodied, pale soft-bodied individuals. So we want to call as tenerals. So if you observe those life cycle of an insect, so every insert is there. So caterpillar first insert is there, second insert is from first insert to second insert. So the newly formed insert or second insert we want to call as newly formed or newly emerged. So later it will become hard. If you observe life cycle of any insect, then you can definitely know about these concepts. Even our human beings also like that only after birth. So in case of a childhood stage, very soft body is there, means very delicate bones. Softer at the end of at the age of 20 to 30 ages. So these bones will harden, it will become hard. Okay.
Yes, sir.
The father of insect morphology or is Snodgrass. You can remember those now. Snodgrass. Yes, if you have any doubt, you can ask. No.
Yes, sir.
Just you can 1 minute. Not so in case of eye care examination, some disturbance is there. Okay. So in case of ICR examination, so they will ask question from cuticulum. So chitin it is absent. So like that only chitin it is more present or more abundant in for N-acetyl glucosamine. So they will ask those type of bits only. Otherwise they will ask about sclerotin arthropodia. So you can remember those points. Snodgrass. Okay. Okay, that's all about today's topic. Again, we will meet on Wednesday. Thank you.